Okay. This is our slideshow on protein synthesis. Protein synthesis has two phases that help make a protein.
Okay, so the first phase is transcription. And transcription is basically when the RNA polymerase, which is made of enzymes, it binds up with the DNA at complementary sequences of nucleotides. It attaches to those, and then it starts transferring the genetic information over from the DNA to the RNA. And then the RNA, once it's done getting the genetic information, detaches from the DNA and leaves the nucleus. And then it goes to attach to a ribosome, which is when translation starts.
The first step in translation is when the strand of RNA enters the cytoplasm of the cell, and it then becomes an mRNA because it's small enough to slip through the nucleus, and it's done copying the genetic code of the DNA. Once it attaches to a ribosome in the cell, then like that, then tRNA cells start matching up anti-codon sequences to codon sequences. As well, that every tRNA that comes, it carries its one amino acid that it was carrying, and it connects it to the polypeptide chain. Meanwhile, the ribosome keeps on going until it hits a stop codon sequence. After these steps, when it hits a stop codon sequence, it will stop scanning the code. After these steps, the polypeptide starts acting like a functional protein, and the whole process starts over again. And this is because if after the protein is made and it's in an unbound ribosome, then it will stay in the cell and report to do whatever it needs to do in the cell. But if it attaches to a bound ribosome in the rough ER, then it will get transported into the Golgi apparatus after that with help from the vesicle. And after the polypeptide is done getting synthesized and coming into creating a protein, the vesicle will send it outside of the cell, and that's what could happen if it does not stay in the cell and does its job. And that is our slideshow on protein synthesis.
Thank you, everyone.
Thank you, everyone who's watching. Oh God, stop.